Okay, everyone, it's our great pleasure to have Carlos Brody here today. You're welcome. So, Carlos, like many great neuroscientists, got his start in physics. He studied physics at Oxford. Physicists in the group. Then he did his PhD at Caltech with John Hopfield, one of the giants in the field of computational neuroscience, where he published some classic papers on synchrony, and the What is a Moment series is actually a very good series of papers for the grad students in the audience who haven't read those. After he did his PhD purely in computational work, he went on to do a postdoc in Mexico with Rinaldo Romo to study working memory, and in particular parametric working memory in the somatosensory system of primates. And there, I think this has been fundamental to the way he thinks and has done science since then, studying simple models of how animals and nervous systems integrate sensory information, how they hold this in memory, and how they make a decision about this, using really elegant, simple interpretable models, which I think is one of the hallmarks of his work. After his postdoc, he started his own lab at Cold Spring Harbor, where he was again in the climate of studying decision-making, but now in rodents, and I think this inspired him to, when he moved to Princeton, he said, "The heck with this, I need to do experiments in my own lab." And now he's got a really elegant lab where they do experiments investigating the same themes of memory, sensory accumulation, sensory evidence, decision-making, and simple computational frameworks to really link the two together. So he's been well-funded throughout his career by the NIH, recently the Simons Foundation, and he's been a Howard Hughes Medical Institute investigator since 2008. And we're very excited to hear what you're up to now. Thanks, Carlos.
All right. Thank you very much. Thank you all for coming, and thank you, Bilal and Garrett, for inviting me. It's a pleasure to be here. So I'm going to tell you about what we're interested in my lab. My apologies to that side of the room. I'm going to be pointing on this screen. So we're interested, as Bilal mentioned, in what we like to call the neural architecture of cognition. So what do we mean by cognition? We mean processes such as these. Working memory. How do you keep in mind some information? How do you manipulate that information over a span of a few seconds? Planning. How do you use knowledge of relationships between actions and their outcomes in order to decide what to do? For example, if you're playing chess and you know that moving a pawn would put pressure on a bishop, how does that inform you in terms of what move you're actually going to do? What psychologists like to call executive or cognitive control. And I like to describe that as your ability to control how information gets routed within your brain. For example, I could tell you right now, oh, today red means go and green means stop. And you could go out and you could drive like that. And it would take a little bit of effort, a little bit of mental effort, but you could do it. So how is it that just by deciding that you're going to follow that instruction, you can reroute red from going to the brake pedal to now red goes to the accelerator pedal? How do you control routing of information in your brain? And decision-making, evidence accumulation for decision-making. How do you accumulate evidence when you're making a decision? How do you accumulate evidence over time for or against different options? And how do you use that accumulated evidence to eventually commit to a decision and implement it? These are the kinds of things that we're interested in. And I got to that interest, as Bilal described, partly by being in Rinaldo Romo's monkey lab, where they were studying these questions. And when my lab first started doing experiments on these kinds of questions about 10 to 15 years ago, studies of these kind of cognitive processes, sometimes referred to as higher cognition, which is a term that I like to use because it makes it sound really sexy, although I don't really know exactly what it means, but I'll still use it. I'll call it higher cognition, makes it sound interesting. So studies of this kind of higher cognition at cellular resolution, if you wanted to know how did single neurons or many single neurons participate and represent this kind of information. These studies were mostly done in non-human primates. And the general idea was these are very sophisticated processes. You need a sophisticated brain in order to implement them and study them. And one of the difficulties in using non-human primates, as people that do this kind of work know very well, it's extremely valuable work, but it's really difficult. One of the major difficulties being that it can take a long time to train an animal in a particular behavioral task that you've designed carefully to isolate a particular cognitive process and allow you to study it. it's not atypical to see training times of 6 to 12 months per animal. And so as you guys know, if you've read monkey papers, what you typically see are papers that have 2 animals in them, right? And typically one monkey was great and the second monkey was kind of okay, but it's the backup monkey to show that it wasn't just in a single animal. And so that makes the cycle of you have an idea, you want to test it out, and you find the results, and then that refines the idea. That cycle, that experimental hypothesis cycle, can be really slow and really hard. And so when I was at Cold Spring Harbor, I had 2 neighbors that had huge influence on me, and they were Tony Zader and Zach Mainen, and they had started trying to treat rodents the same way people were treating monkeys. And by that we mean trying to do kind of rigorous psychophysical studies of them, rigorous behavior, and trying to address the very same kind of questions. And so I decided to join them, and together we pushed a lot on that. The reasons why we were training to rodents, initially with rats, is that they're comparatively small, comparatively inexpensive, you can get good behavior out of them, and they're amenable to modern molecular tools. So here's a picture of one of the walls in the current behavioral training room in my lab. These are 9 different rigs. This is a computer rack, so a person is about that tall, just to give you a sense of scale. In total in the lab, we now have 36 rigs, and we run 9 shifts per day per rig. Okay? So that means that we're training a total of 324 individual rats per day. And that's what we can do because of the relatively small, relatively inexpensive. That gives us about 1 million behavioral trials per week. So that gives us a lot of information to do careful statistical analyses. That in itself is extremely useful. A typical student or postdoc in my lab supervises 20 to 30 animals. If you're supervising 20 to 30 animals, there's no way you can train them the way people train monkeys, which is to sit with each one individually and kind of see what it does. An almost artisanal kind of training. You can't do that on that scale. So the only way to do it is to develop computer code that will do the training for you, that will take the animal through all the shaping steps that you want for a particular behavior. And the advantage of doing that is that once you've got the code, well, you can just copy it as much as you want, and you can parallelize as much as you want. Okay? So the tasks that we study in the lab, they typically take about 4 months of training per individual animal. But if we take 40 out of these 324 slots and dedicate them to a particular task, which is a typical kind of thing that we do, then on average, we get 1 trained animal out every 3 days. And that's a completely different world to having a couple of animals per year, right? It lets you try out many more things. And so the idea is that we'll use this as a platform to make that hypothesis experiment cycle to try to make it faster and make faster progress in a way that should be complementary to the studies that are being done in the primates. So together with Zach and Tony, we were part of labs pioneering, studying neural circuits for cognition in rodents. Of course, there were many other labs that were doing this before us. Kind of like the thing that we pushed on was this idea of treating them almost as if they were small monkeys, trying to address the very same kind of questions with the same kind of behavioral and quantitative rigor. Since we started, there's many more labs that are doing that. It's a big wave of people doing that, some people here, and there are many, many labs participating in that. My own lab, our particular interest and focus has remained on these more complex, what I like to call the higher cognitive processes that I described in the first slide. And these, in practice, are typically turn out to be tasks that require a lot of time for training an individual animal, and therefore they're tasks where this kind of setup, where we do this semi-automated high-throughput training, can be extremely useful. So that system, that high-throughput training, has really allowed us to push the envelope on what we know rodents are able to do. So for all 4 of the processes that I described in the first slide, all projects in all 4 of these started with a paper that showed either that rodents were capable of something that people didn't think rodents were capable of, or that they do it in a way that is very similar to humans and monkeys in a way that people didn't think was possible. So we're really pushing the envelope of how far they can go. We don't yet know how far we can push it, right? There are going to be limits to it. But so far, it's being pushed in a very interesting way, at least for us. So we do 300-odd behavioral sessions per day. In contrast, when we do electrophysiological recordings, once we go beyond behavior and try to get into the brain and do recordings or perturbations, each of the recording rigs is a We have 4 recording rigs, and those require much more manual intervention, so individual people sit with each animal during the recording session. So as a result, we only get about 3 to 4 electrophysiological recording sessions per day. So there's a 2 orders of magnitude difference between these. And that has actually felt very painful over the last 10 years, right? Because the whole project was, oh, we're going to move so fast. That cycle is going to be so incredibly fast. And actually, we have a bottleneck there where it's not so fast. So we want to work on that too. And in particular, using some technology that Ala Karpova's lab at Geneva has developed. And there's other groups that are doing this kind of thing. They're really helping us for that. So these are wireless electrophysiological recording and wireless optogenetic perturbation. The recording in particular is now through a company called Spike Gadgets that originally came from Ala Karpova's lab. And the reason that these wireless systems are so useful and important for us and combine so well with our high throughput training system is that because with the wireless systems, we do not need a specialized rig to do recording or optogenetics. And so that means that we can really do it in all of our behavioral training rigs. Any one of our rigs now becomes an ePhys, an opto rig. So we're really pushing on that so that we'll be able to get 324 electrophysiological and optogenetic sessions per day and really match these 2 numbers and hopefully move really as fast as we were hoping to do when we started on this. So that's telling you a little bit of the background, a little bit of the kind of framework in the lab, a little bit of the simple technology that we use. But to get into the science. I described some of the processes that we're interested in, and the regions in the brain that we study, we start by trying to guess which regions in the brain are going to be important. And it turns out that for all 4 of these, there's an overlapping set of brain regions that are relevant. These are the initial guesses, right? It's not that we're limited to those. We want to explore more and try and understand what other brain regions are involved. But these are the initial guesses. And because they overlap across all 4 of these, studying these 4 different processes becomes really useful. Because it gives us, each one of those gives us a complementary view on these different brain areas. And so we can get understandings that we wouldn't have gotten to if we had focused on a single process. And so what I'm going to do today is tell you a story from 2 of these projects. I'm going to tell you a story from decision making and from working memory. And you'll see that the 2 combined in a particularly interesting way. And I'm choosing these 2 because they illustrate this idea that by coming with different projects to the same brain areas, we can gain understandings that we wouldn't have gotten otherwise. So I'm going to start with evidence accumulation. And by the way, I'd like to emphasize, please interrupt me and ask questions as much as you like. I'll keep an eye on the clock. And I know you guys have to leave exactly at 12:15. I'll be responsible for the clock, so don't worry about it. If it gets late, I might say, if you ask a question, I might say, you know what, I'll leave that till later. But don't worry about the clock. I'm responsible for that. You please ask as many questions as you like. OK, so decision making. I'm going to start by pointing out perhaps the most commonly observed behavioral observation in decision making, which is simply the fact that when you've got a really difficult decision to make, it takes you a long time. Whereas when you've got a really easy decision to make, you can do that very quickly, right? If you're deciding between 2 awesome grad schools that have accepted you, it takes you a long time to make up your mind. If you're deciding between a fantastic grad school and a horrible grad school, you decide really quickly, right? So that has been observed not only anecdotally in your daily lives, but also in the lab in a whole host of various types of decisions, social decisions, perceptual sensory decisions, very famously by Bill Newsom and colleagues, economic decisions, gambling decisions, memory decisions, visual search decisions, value-based decisions, all kinds of decisions. And because it's being observed so commonly, it's thought that there's a common core process that sometimes goes under the name of noisy accumulation that perhaps can account for this observation across all of these different decision-making domains. And the idea behind it is very simple, really. So I'm going to illustrate it now. Perhaps some or many of you have already seen this before. But imagine that you're a subject in a lab, and you're going to do a trial where you're going to get shown a stimulus, and based on that stimulus, you have to decide between orienting to the right or orienting to the left. Okay? And now pretend that somewhere in your brain, there's some abstract variable, an abstract scalar variable, that we're going to call the evidence accumulator A. And where exactly it's represented in the brain and how it works in the brain, that's going to be some of the main questions. But for now, pretend that that scalar exists. And pretend that at the beginning of each trial, the value of that scalar is 0. And then as the trial unfolds and you're seeing the stimulus, every time that you see something that makes you lean towards going right, you add something to A. And every time that you see something that makes you lean towards going left, you subtract something from A. And you just keep going like that over time until you get to the end of the stimulus. And at that point, you have to make a decision. And the idea is that what you do is ask, what's the sign of A? If by the end of the stimulus, the sign of A is positive, you go to the right. If by the end of the stimulus, the sign of A is negative, you go to the left. Super simple model, right? But you can already see that it accounts for this observation. If the evidence is weak, namely what you're adding or subtracting from A at each time step is small, then it's going to take you a long time to move away from the decision border at A equals 0. If by the end of, say, a whole 2 seconds, you're not far away from the decision border and there's noise, that's what's going to mean that it's a difficult decision. It's hard to tell on which side you should lie. Right? Whereas if what you're adding or subtracting from A is at each time step is really large, you're going to move far away from the decision border very quickly. And so that's going to be an easy and a fast decision. So super simple model, accounts for a ton of behavioral data. Of course, people love it. Right? And it showed up in the behavioral literature about 50 years ago, and it's been very, very widely applied. Now, in this model, the idea is that on each individual trial, the trajectory of A as a function of time could be quite noisy. But if you averaged within classes of trials with a particular strength of the evidence, you should see the trends that I described before. Namely, if the evidence is weak, on average, A should grow relatively slowly, so a shallow slope here. Whereas if the evidence is strong, the evidence should grow much more rapidly. Okay? And starting in 1996, Mike Shadlin and Bill Newsom did a series of experiments that were really seminal experiments. They started a whole school of many other people following this, where what they did was they had monkeys doing a perceptual decision task, a sensory decision task, where they could control how strong the evidence was on each trial. And they recorded from a particular part of the brain that I'm going to refer to as PPC. The particular area in the monkey brain that they recorded from was lateral intraparietal cortex, LIP, which is part of PPC. Here I'm going to refer to it as PPC. They recorded from neurons in PPC while the monkeys were making up their mind before they reported what the decision was. And what they found there were firing rates that ramped, kind of like what you see here. And critically, when the evidence was weak, the slope of the ramp was shallow, it was small. But when the evidence was strong, they ramped much faster. Okay? Just as the kind of picture that you have over here. And that similarity led to the proposal that PPC firing rates encode the value of this abstract variable A. And that was a critical finding because this model, this noisy accumulation of evidence model, had been around for decades, tons of behavioral papers about it. But this was the first time that anybody had seen a signal inside the brain that corresponded to this putative variable A. So super exciting thing, set up a whole school of thought. And really, we're kind of like following in their footsteps. So their first experiments, as I said, were in PPC. And the idea was there's a one-to-one relationship between firing rates in PPC and the value of this abstract variable A, the evidence accumulator. But there's been recordings in several other brain regions since then. For example, the frontal eye fields, where the same phenomenon was found. So for weak evidence, shallow ramps. For strong evidence, much sharper ramps. And these two cortical regions are thought to be key areas for this process of evidence accumulation. But the same kind of qualitative conclusion has also been seen in other parts of prefrontal cortex, in subcortical areas like the striatum, and areas in the brainstem like the superior colliculus. If you look at a subset of the connections between these different brain regions, you basically see that everybody's connected to each other. And of course, I'm not even putting in other brain regions where which gives rise to indirect connectivity. And this, figuring this out or looking at making these observations has been happening for the last 20 years or so in multiple labs. And the similarity in what's observed across all of these different brain regions has led to the proposal that maybe the mechanism for this, the mechanism for how evidence is added over time, how it's integrated over time, maybe it's a diffuse circuit mechanism widely distributed across the brain. We don't know. The way I like to refer to that is essentially to say that we don't really know anything yet about the circuit mechanisms, right? That's what, to me, this proposal really sounds like. So when we started working on this, some of the questions that we thought were really basic, So when we published our first paper 5 years ago, 6 years ago now, some of the basic questions that were still unanswered were questions like this. Which of these brain regions is necessary or not necessary for this evidence accumulation process? Not known. Where is the evidence accumulation computed? Is it in any one of these brain regions, or is it in somewhere else that hasn't yet been recorded from? Not known. How is the evidence accumulation computed? What is the actual mechanism that leads to that accumulation? Well, if you don't even know that you've recorded from the place where it's happening, of course you don't really know precisely how it's happening. It's really hard to have a model for that. So our thought was, well, let's see if we can use our rats in our high throughput training system to do this faster cycle, and maybe we'll provide some information that's complementary to what's being done in the monkeys and unravel the mechanistic circuit logic, help to unravel that. That was the idea. And when we set out to do this, the first question that we faced was, well, can rats accumulate evidence over time for decision making on timescales that are similar to what had been seen with primates, both human and non-human? Right? Maybe rats just can't do this. And in fact, some of my monkey researcher friends, that's exactly what they told me. You're an idiot. Rats aren't going to be able to do this. You shouldn't even try. So that was a critical question to address. Right? Can they do it? And so the first paper that we worked on for this was a paper where we set out to design a behavioral task that would try to get rats to do this process and that would allow us to quantify or do quantitative analysis on how they're doing the task that we've put in front of them to try and answer this question. Okay. So that's what I'm going to describe to you now. So in any one trial of the task, our rats are in a behavior box where they face 3 nose ports that are in front of them, in a wall in front of them. And a nose port is a little place that they can stick their nose into, and we have an infrared beam across it so we know when they put their nose in there. And I'll take you through one trial in the task. The very first thing that happens is that we have an LED in the center port that turns on. And that's the signal to the trained rat, come and put your nose in here. And the rat is trained to keep its nose in that center port until we turn that center LED off. We call this the nose fixation period. So they put their nose in there, and then we have a speaker to their right and a speaker to their left, and we play randomly timed clicks from both speakers going at once. Click, 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 click. And then when those clicks, those 2 trains of clicks, simultaneous trains of clicks end, we turn the center light off so the rat is free to withdraw, and it should choose to go either to the right or to the left. And if it goes to the correct side port, it gets a drop of water. We keep them a little thirsty. That's what they work for that reward. If it goes to the incorrect side port, then we give it a white noise and a timeout, and then the next trial starts. So what defines which of the 2 side ports is the correct port, the one that is seeded with water? It's which of these 2 sides, right or left, had played the greater total number of clicks. Okay? That's what tells the rat which way to go. So why do we give it that particular stimulus? Well, the reason that we chose this kind of stimulus is that if you're going to solve this task, if you were to do this task 100% correct, get it right on every trial, then what you'd have to do would be to add clicks up over time, add auditory clicks up over time from the 2 sides in order to be able to compare the 2 totals and say which one had the greater total. Right? Adding auditory clicks up over time, that is the same as accumulating sensory evidence over time. Right? So we're trying to set up a task where the optimal thing to do is to accumulate sensory evidence over time to follow the cognitive process that we want to study. Now, nobody does this task 100% correct. Not rats, not humans, not monkeys, not anybody. So there's a lot of wriggle room in that, well, the rats are doing it, but not quite 100% correct. And we're going to worry about that quite a bit. OK, so in this task, I'm going to skip over the evidence that rats actually are accumulating evidence over time. It's in this paper, and if at the end of the talk you want to ask me about it, I'll be happy to go into it at length. But I want to get to some other stuff. For now, I'm going to ask you to please just go with me. Keep your questions. I know it's an important question, so please do ask me at the end. But for now, imagine that rats are actually accumulating clicks over time in order to solve the task. I want to point out that there are at least 2 very critically different time periods in this task. The first is while the clicks are playing and the rats are accumulating the clicks over time. And you can imagine this as each click, each rightwards click being a +1, a positive delta function, and each left click being a negative delta function. And you just integrate those over time. So while the clicks are playing, you're doing this integration over time. But then once the clicks end and we turn that center light off, that's the go signal. That says to the animals, now you have to decide. And now what has to be computed is not integration over time, but ask, what's the sign of A? Right? Because that's what tells you, should you go left or should you go right? Okay? And the difference between these two time periods is going to become important later, and I'll remind you about it then. So we made these clicks random in time. And if we make them random in time, that means that on each trial, we literally have a lot of bits of information, right? Because the timing of each click is random, can be anywhere, but we know to the microsecond exactly when it was played. And so that gives us a lot of statistical power that allowed us to do a lot of analyses. In particular, the model of the accumulation process that we had a lot of parameters, but with that statistical power, we could have relatively low error bars on each of the parameters and distinguish some of them. And we were able to quantify some things for the first time. And it was also a big part of what convinced us that the animals were actually using gradual accumulation of evidence over time to solve the task. And also, another critical thing that this gave us is that knowing exactly when the clicks played allowed us to build a model that modeled on a moment-by-moment basis how this variable A was evolving over time within each individual trial. Okay. And so in response to a particular set of clicks, we have an estimate of how A evolves. Now, we think the process is noisy. There are also noise parameters in there. So actually what we evolve is not a deterministic estimate of A, but a probability distribution over values of A. That's what we evolve over time. So the equations that we're actually using, for those of you for whom this means something, we're using Fokker-Planck equations to evolve this. And then when we get to the end of the trial, we know what the animal actually did. So for example, if this was a trial where the animal went to the right, we know that that means that this probability distribution must have all its mass on the positive side of 0. So that puts a constraint on this, and we can use the backwards Kolmogorov equations to reverse propagate that constraint backwards to get the most precise estimate at any point in time of what we think that probability distribution of values over A is. Okay? So that allows us to use the exact timing of each click. It's a model that takes into account the timing of each click and the animal's decision to use all the behavioral information that we have to give us that estimate of A, the distribution over A, and how it evolves over time. When we're doing recordings from neurons while the animals are performing the task, then from each individual neuron, we also have an estimate of how the firing rate evolved over time. And we have these two estimates simultaneously. Having those two estimates simultaneously means that at each point in time, over trials, we can build up a map of how those two relate to each other. Right? We simply say, when the distribution of values of A is over here, how much is the firing rate? When it's over here, how much is the firing rate? When it's over here, how much is the firing rate? And so on and so forth. And so that allows us to build up a map of how firing rate of the neuron relates to the value of A. In other words, a tuning curve. If you were recording from a neuron in visual cortex, what are the basic kind of things that you would want to ask in order to build an intuition or a sense of how information is encoded? Well, for example, if you're using oriented bars, you would plot firing rate as a function of the orientation of a bar, right? Firing rate as a function of the variable of interest. Well, in this case, in decision-making studies, before we had this estimate of what A was at each point in time, we didn't really have this horizontal axis. So nobody could really plot that. But once we had that estimate, that allowed us to make these plots and really estimate what these tuning curves are in different brain regions. Okay. So that is going to be a tool that is going to be very useful for us. And I'm going to talk about it, about the results of using it in a few slides. And having all these tools in hand, yes?
What are the error bars in the plot?
What are the
Error bars.
Oh, where are the error bars?
How big are they?
Oh, here they're really tiny because they're averaged over many neurons and they're averaged over many rats. So they're actually only a little bit bigger than these balls.
And how different are individual neurons?
Different neurons are quite different. So I'm going to mention that later. So in particular, within any one brain region, it is not the case that all the neurons behave the same. And I'll come back to that point again. Any other questions at this point? Not right now. That probably means I'm speaking too fast and it's just all gobbledygook. Don't hesitate to ask. OK, so we have a tool. We've convinced at least ourselves and a small fraction of people, or perhaps a growing fraction of people, that rats are accumulating evidence over time as we go. So that means that we can study this process. We have a tool to assess how information is encoded in any neuron that we're recording from. And so now we want to go to that question of what are the critical regions that we should study? Right? So our thinking was that any particular brain region that was causally involved in computing the gradual accumulation of evidence over time should, first of all, carry information about the graded value of the accumulator. Notice that this accumulator is a graded quantity. Okay? And so we're going to use our tuning curve measurements as a tool to help us assess that as we record through different regions of the brain. Second, if that region is involved in computing the gradually accumulating evidence, well, if we silence it, that should have an effect on the behavior, given that the behavior is based on accumulation of evidence. And third, if we perturb it during any part of this accumulation period, that should also have an effect on the behavior. So in practice, what these three considerations boil down to are 1, computing our tuning curves, 2, pharmacology. We inactivate regions pharmacologically and we see if it has an effect. And 3, optogenetics. We inactivate regions optogenetically to give us time precision to look at the difference, to look to see whether this whole time period is relevant. Okay. So off we go. Now we're ready to go into the brain. And there we face this difficult problem of like, nobody's done this in rats before. Where should we go? I don't know. The whole brain is fair game. So we did the next best thing, namely we looked to the primate literature to get inspiration from that. And there are all these regions that had been recorded from in the primate literature. And so we thought, well, we'll start with brain regions in the rat that appear to have similarities to corresponding brain regions in the primate. And so we started with 2 regions, PPC and FOF in the rat. This has a lot of similarities in terms of its connectivity and some of its functional properties with primate PPC, and this has a lot of similarities with the primate FOF. And we started the primate FEF, the frontal eye fields. We call it the frontal orienting fields in the rodents. And we started with these 2 because these were the 2 most prominent regions in the primate literature for evidence accumulation. So let's start with PPC. And so we put tetrodes in PPC while the rats were performing the task, and we recorded from many neurons. And we used our tuning curve tool to try and get an estimate of how firing rates in PPC related to values of the accumulator. And what we found was that there was a relatively smooth relationship between these 2. Okay? So that means that if you know the value of firing rates in PPC, you can read out an estimate at each point in time of what the value of the accumulator is. Okay? Which is essentially the same thing that Mike Shadlin and Bill Newsom had proposed all the way back in 1996. Right? We used a very different approach, and now we're showing it in a species that they had not studied. We showed it in rats, and we think that we showed it in a more rigorous way. But the main message is essentially the same one. So sometimes I like to say, all that work just to show that those guys were right. But then we did something that they had not done, which was inactivate PPC while the animals are doing the task, just to probe causally what's going on. And that gave us a big surprise. So here what I'm showing you is the percentage of trials in which the animal went to the right as a function of total number of right minus total number of left clicks that were played on each trial. And black is the control. So that's if we just regular trials, we don't do anything. And then we used mucimol, a GABA agonist, to inactivate on one side. So if we inactivate on the left, that's the green curve. If we inactivate on the right, that's the red curve. If we inactivate on both sides simultaneously, that's the blue curve. And essentially, you don't see anything. So then we doubled the dose, and we didn't see anything. So then we quadrupled the dose, and we didn't see anything. And then we octupled the dose, and we didn't see anything. And we thought, we're just doing things wrong. And so we added a different type of trial that was intermingled with these. That was a control type of trial. And we found a big effect in those controlled trials. So we know that our mucimol is working. There's just no effect in the PPC. So that rules out the PPC as being a main contributor to this computation. Right? Maybe the PPC is part of a broader redundant network. That could be, but it certainly can't be kind of a key central node in this process. Right? We'll come back to PPC later in the talk. And these experiments inspired some very similar experiments in monkeys. So Alex Huck's group at UT Austin then, using the same task that the monkey researchers had used, inactivated PPC in the monkeys and found essentially the same result. You inactivate LIP, there's no effect. So now we know that our result is not specific to rats. It's a more general thing. So we moved away from PPC, and then we turned to FOF. And one of the first things we did there was inactivate FOF, hoping to see some kind of effect. And then we did see a big effect. So if you inactivate the right FOF, the animals go much more to the right. If you inactivate the left FOF, they tend to go much more to the left. So that tells us that the FOF is playing some role in the task. It doesn't tell us which role it's playing, right? It just tells us that it's in there somewhere. So then we did electrophysiological recordings in the FOF. And here in red, I'm showing you the data from the FOF, and in faint purple, the data from the PPC. And what you can see is that the FOF has a much more step-like tuning curve. Okay? And that was extremely interesting to us, in particular because of where the border of the step was. And the border of the step was exactly at a equals 0. Okay? Now I want to remind you, a bigger than 0, that means go to the right, whereas a less than 0, that means go to the left. Okay? So what this was telling us was that in the FOF, if you know the value of firing rates in the FOF, you don't have a good estimate of the graded value of the accumulator. But you do have a very good estimate of what the sign of the accumulator is. Okay? So that suggested that maybe the FOF was more involved in this process than in this process. So it's part of the task, it's part of the decision-making behavior, but it's not a part of the gradual accumulation of evidence over time. So coming back to Elia's question, here I'm showing you averages over neurons. And different neurons can be very different. So some neurons in the FOF are as smooth as the neurons in the PPC. So that's something to keep in mind. Another thing to keep in mind is that even in this average, this red line, I'm talking about it as if it were a perfect square step function. But you can see that it's not. Right? There is some graded information in there. So these electrophysiological results suggested that the FOF was more involved in this than this, but certainly didn't prove it. So to try and get a better look at that, we then turned to optogenetics, using halo rhodopsin to inactivate neurons. And in particular, we're going to use it to inactivate the FOF unilaterally. And for us here, the big advantage of using optogenetics was the time resolution. You can silence neurons within about 16 milliseconds. And when you turn your silencing laser off, activity comes back in about 60 milliseconds. Right? So that meant that we could silence different subperiods of the trial. And so we did that, and I'm showing you the results of that experiment here. So on the y-axis, I'm showing you the percentage ipsilateral bias that was caused by inactivating the FOF. And the horizontal axis, the extent of each horizontal bar, tells you when the FOF was silenced. Okay? So what I'm showing you here is that if we silenced the FOF before the click started, it had essentially no effect. If we silenced the FOF during the first half of the clicks, it had no effect. If we silenced the FOF once the motion had started, it had no effect. But if we silenced the FOF during the second half of the clicks, just before this go signal, that's when we did see a big effect. Okay? And so then we did the experiments again, but now having the time that we were using. So now looking at either the last 250 milliseconds before the go signal or 250 milliseconds before that. And what we found was that once again, only in the very last few hundred milliseconds before the go signal is when we see an effect of perturbing the FOF. Okay? This is exactly what you'd expect if the FOF was involved in this process, but not this process. If you perturb during the accumulation process, you don't see anything. If you perturb when the animals are trying to compute or use the sign of the accumulator, that's when you see an effect of perturbing the FOF. So that suggested to us that indeed, as the electrophysiological results had hinted, the FOF is more involved in this than this. And there's a question here.
Wouldn't this be related to motor planning? Motor planning?
Absolutely. I mean, the clicks are coming in and you're told, make a decision. That's when you are committing either to go left or right, and you make your motor plan to go either left or right. Yeah. So these results tell us that the FOF, they don't tell us that the FOF is involved in computing the sign of A. They tell us that the FOF is involved in either computing it or representing it or using the sign of A. So for example, if the FOF is necessary to make the left versus right orienting motor plan, then this is exactly what you would see. Yes?
I presume that the tuning curve on the top is calculated from the whole time period?
So one of the things that we did was calculate tuning curves at each time point.
Yeah.
And in both the PPC and the FOF, it turned out that different time points were very, very similar. So that's why I'm showing you At that point, we averaged over time, and that's what I'm showing you here.
The curve doesn't stretch.
The tuning curve does not stretch. It did not have to be that way, but that's how it turned out. OK, so that told us that the FOF was very interesting. But again, it suggests that it's not part of this accumulation process, of this integration over time, which is the thing that we were trying to hunt down. So then we turn to the superior colliculus, and I won't take you through the data in the superior colliculus, except to say that it looks very similar to what we found in the FOF. So we think that the superior colliculus is also more involved in this process than this process. And then we turned to the striatum, in particular to the anterior dorsal striatum. If you're a striatum aficionado, you will distinguish between dorsomedial striatum and dorsolateral striatum. The place that we got to it because we were following projections from the FOF into the striatum. And it turns out that the projections kind of like land right on the border between dorsomedial and dorsolateral. And so that's where we went. And so that's why we don't call it either dorsomedial or dorsolateral. All we really know is that it's anterior and dorsal. So hence the name that we used. And we did the same things that we'd done before. We record there. So this was Mikhail Yartsev in the lab. And we found a relatively smooth tuning curve, kind of like in the PPC. So if you know firing rates in the FOF, you can read off at any point in time the value, the graded value of the accumulator. But then unlike the PPC, when we did the pharmacology, there we found a big effect. Okay? So certainly suggesting that the ADS is involved in some way in the task. And then when we did the optogenetics, we found something that, at least to us, was extremely interesting. So here in red, I'm showing again the data from the FOF. And once again, I'll point out that in the FOF, only immediately before the go signal do you see anything. And in green, I'm showing you the data from the striatum, from the ADS. And what we found there is that in the striatum, no matter when during the accumulation time period we perturbed, we found an effect on the task. Okay. So these are the three things that we had set out to hunt for, right? Silencing affects the accumulation of evidence behavior. The firing rates represent the graded value of the accumulating evidence at each point in time as it's accumulating. And perturbing at any point in time has an effect on the accumulation process. So we were very worried when we ruled out the PPC and the FOF. We thought, well, who knows how many years we're going to spend hunting and ruling regions out. Right? So it was actually quite a relief to find at least one region that matched our criteria, and that we can now say, we think that this is at least one node in the circuit of nodes that are involved in computing this accumulation of evidence over time. So we're very excited about that. We're not excited about this message. So we're very excited about that. And we're also excited that we think that the FOF and the SC are involved in this process, because then we think that we've got a set of brain regions that represent the entirety of the process, all this stuff and this stuff. And we're really interested in, of course, how does this graded signal get turned into a binary signal, the categorical go left versus right signal. And we think that the interactions between these might tell us a lot about that. So what we're doing now is doing simultaneous recordings in pairs of regions, and also simultaneous recording on optogenetics in pairs of regions. In this cartoon, I had 3 regions simultaneously, but really we're going 2 regions at a time to try and understand how these regions interact with each other, both through looking at correlations between the recordings and the effect of perturbing one region and seeing the effect on firing rates on the others. Now, I want to point out, we think that the ADS is a node in the circuit that computes accumulation of evidence. We're very excited that we've finally identified one. But we don't necessarily think that it's the only node in that circuit. And so to probe that, one of the things that we did was inject retrograde beads into the ADS and just look in the rest of cortex, ask where do those beads go. And we had gotten to this particular part of the striatum, the anterior dorsal striatum, by following projections from the FOF to the striatum. And so we weren't at all surprised to see that some of the beads, some of these retrograde beads, ended up back in the FOF. But what we were surprised by was that the strongest signal, most of the beads, ended up in a region that's anterior to the FOF, 2 millimeters away, which in rat scales is a totally different brain region. So this anterior M2 region is now of huge interest to us. We're very excited about that. Nobody has studied this region whether in rats or in monkeys, in the context of evidence accumulation over time. And we're only just starting the experiments there. At least I can tell you that mucimol in this brain region has an effect on the behavior. So we know that it's not going to be like the PPC. But what its role is, that we don't know. That's what we're pushing on. And the biggest point, we think, is that having identified one node, that was the key thing. Because now we can follow the anatomy and unravel the whole circuit. Right? Here, we're following it upstream, but we're also going to follow it downstream to try and understand how the whole circuit works. At least that's the plan. Now, I'm going to turn back to the PPC that a few slides earlier I said, oh, pharmacologically, when you activate it, it has no effect. Meh, forget about it, right? I put this big red line over it. But it's kind of mystifying, right? Because another thing that I told you was that the PPC did have a really good representation in its firing rates of the value of the accumulator. So what is that representation doing there? And if you look at connections between the PPC and the rest of the circuit, well, it's pretty strongly interconnected with the rest of the circuit. So what role does it have? And that's where this other project that we'd been working on gave us some really interesting hints. So I'm going to switch to a different story. And just to tell you where I'm headed, the results of this told us that, at least in the tasks that we're studying, PPC mediates long-term, where by that I mean effects over multiple trials, of sensory history. The history of the stimuli that the animal has received have an effect on the animal's behavior, and they do that through PPC. So let me take you through the experiment where we found that. And in this experiment, again, in each of the trials, the rat faces the wall with the 3 nose pokes, and it's trained to put its nose into the center poke. But in this experiment, once it puts its nose into the center poke, we do the following. We first play a stimulus, a pink noise stimulus that we call SA. And then there's a delay that can be quite long. We've taken it up to 12 seconds. And then we play a second stimulus that we call SB. Okay. And then there's a little delay and then a little tone beep, that tells the animal, OK, now you're free to go. And now what the animal has to do is respond based on which of these two was louder, the first or the second one. Okay. In order to do that, you have to remember the loudness of SA throughout this delay period so that you can compare it to the loudness of SB. Right? So it's a working memory task in that sense. You're keeping this information in mind. And we call it a parametric working memory task because the loudness of SA is a parameter. It's a continuous parameter. So that gives us something to plot against, which can be extremely useful in the analyses that we do. Okay. Now, if as you go through this task, if you ask how difficult is each of the trials, well, that depends on how close to this diagonal SB equals SA line you are. Right? If SB is equal to SA, then it's impossible to say which of the two was louder. But if you're very far away in this SB versus SA plot, if you're very far away from this diagonal line, then it's very easy to say what the trial was. And on different trials, we use different pairs of SA and B and SB to cover a big chunk of this stimulus space. And these are kind of the 8 standard trial types that we used. And the little number here is telling you the % correct trials that the animals did in this experiment. And you can see that they're doing pretty well, right? Above 80% correct on all of them, and on some of them, quite a lot higher. Now, as I mentioned, how close you are to this diagonal line tells you how difficult the trial is. So we also had another subset of trial types that spanned this line horizontally from here to here to give us an estimate of the finesse with which the animals can respond and distinguish these two stimuli. And so if we plot the fraction of the time that the animals respond by going to the right as a function of SA minus SB, we get these curves, which are like the psychometric plots that I've shown you in the decision-making task before. The ideal performer would look like this. Right? Anytime that SA is bigger than SB, no matter by how much, even if by very little, go to the left. Whereas, if SA is less than SB, you go to the left all the time, whereas if SA is bigger than SB, you go to the right all the time. Right? This is the ideal performer, and rats, of course, are not ideal, like any observer, any biological observer. Now, one of the things that I didn't tell you is that in this experiment, we were particularly interested in the role of PPC. And we were interested in the role of PPC because there's a lot of rodent literature talking about PPC and its role in short-term memory. So we thought, well, here's a working memory, short-term memory task. It should play a role. what we expect is that if we inactivate PPC, this should impair the animal's ability to perform this working memory task. And so what I hadn't told you is that these data are from animals that had an optical fiber implanted into them, two fibers actually, bilaterally in PPC. And we had also injected a virus to express halorhodopsin so that we could inactivate PPC. And these blue lines are from the subset of randomly interspersed trials in which we had kept the inactivating laser turned off. But we could also turn the inactivating laser on. And when we do that, what we found was that all of the rats looked more like the ideal performer. They all got better. So what we expected is that it would disrupt their performance, and instead they got better. So this was highly mystifying, not what we expected at all. When Athena Krami, the postdoc who was doing this work, who now has her own lab in London and is looking for postdocs, and she's awesome, so if you're looking for a postdoc, first come to me, but she's great too. So when she did the electrophysiological recordings doing this task, here's an example of a typical neuron that she recorded while animals were performing the task. And the different shades of brown in these lines tell you the different values of this first stimulus SA. And what you can see is that during this delay, while the animals are remembering SA in order to compare it to SB, there's essentially no difference between these. Right? So there's no information in PPC firing rates about the value of SA during this delay interval. But as she kept recording into the intertrial interval, so the animals make their response around here, and they've either received a reward or not by about here. And so after that, once this information is totally irrelevant, the trial is over, then there was a lot of information in the firing rate of this neuron as to what the value of SA all the way back here, one trial ago was. So that was a huge surprise. What the heck is this information doing here? The animal doesn't need it anymore. But that got us into thinking, well, maybe we should analyze our behavior and analyze our data, not only in terms of the current trial, but also in terms of previous trials. Okay. And so we set up a very simple model in which we take the values of SA and SB, multiply them by parameters, add them up, also add a bias parameter, and put that through a logistic to produce a probability of the animal going right. That's a super simple model of the behavior. And then we add terms that look into history, into previous trials. Previous sensory stimuli, previous choices, previous rewards, 2 trials back, and so on and so forth. And there's many different combinations of how you could do this and how many trials back you could go. We did a lot of them. Here I'm showing you the particular formulation that had the best prediction of cross-validated data. And if you look at the values of these weights, of these parameters, those are, when you fit this to the data, those are a measure of how much each of these parameters affects the animal's behavior. So if this is very big, that means this has a big effect on behavior. Say if these were 0, that would mean that the sensory stimulus 1 trial back would have no effect on behavior. And what we found was that sensory history weights were significantly different to 0. So previous sensory history has an effect on behavior. And if we take this model and we just predict data from it, of course, we had fitted to the data, so it's no surprise that it predicts the data reasonably well. But then we can also fit the model to the subset of trials where that inactivating laser was on. And when you do that, what we found was that the sensory history weights all got significantly reduced, whereas all other weights, for example, for previous correct side or previous reward or the current sensory stimulus, they were unaffected. So it's as if the PPC was selectively reducing the effect of sensory history on the animal's behavior. And when we keep these parameter values, so when we use these yellow parameter values, but keep the blue parameter values from all the other weights, so we only affect sensory history, then we found we could predict that performance improvement very, very well. Okay. So what's going on? What's going on, it's as if the animals use previous sensory history to guide their behavior. But in the lab, each trial is totally independent from previous trials. So that is a suboptimal strategy to use, right? And we speculate that in the wild, of course, using past sensory history to guide your behavior is a good idea because there are long time correlations in the wild. But in the lab, we artificially eliminated all of those. And so the animal is maybe still trying to use them. And when we stop it from using them, that's when its behavior improves relative to what's being done in the lab. Yes?
So, 20 years ago, there were papers by Gallisol showing that the animals in rats specifically in sort of mice experiments can adapt their time scale to the actual time scale of the experiment. Is there any evidence of that, right?
No. So these are very well trained rats. These are, say, animals that have been doing this experiment stably for 6 months. So we don't see any evidence of them having. You'd imagine that we could train this out of them, right? Because this is a suboptimal thing to do, to use a sensory history. And we also looked at, once the animals are very well trained, look at the first month and look 5 months later, is there any difference? And we haven't found any difference. So this particular thing, we have not been able to train it out of them. I'll also point out that in the subset of trials that we're using, for these psychometric curves, getting rid of sensory history predicts an improvement in behavior. But there are other trials where getting rid of sensory history predicts an impairment of behavior. And that's exactly what we see. Okay? So it's not just that inactivation leads to improvement in behavior. Inactivation of PPC leads very specifically to behavior that looks as if sensory history effects had been eliminated. So what I've shown you is that PPC represents sensory history, mediates the effects of sensory history and behavior, and people are standing up because you got to go, and I got 3 minutes. Why is that result interesting? Because there's such a large literature on sensory history effects. All of Bayesian perception is about using sensory history to guide how you behave now, but nobody had any clue as to which regions in the brain are involved. And as in the decision-making task, now we have a foothold, right? We don't think the PPC is everything, but we can go from the PPC, where it projects to, what projects to the PPC, and try and unravel that circuit. And when we look back at the decision-making task, at the click accumulation task, and now looked at sensory history effects, we found those there too. Right? So this is the case of the working memory project telling us something about the role of PPC in this other project that we would never have found if we had stayed within this one. So people need to leave, so I'm going to wrap up. I'm merely going to tell you that we're also looking at other parts of the brain. I've spoken mostly about looking at effects of whole regions and about behavior at the level of the whole animal. But of course, we're also very interested in how networks of individual neurons drive all these phenomena. And so we're interested in working at this scale and spanning the scales from microns, individual neurons, to millimeters, to centimeters. And I don't really have a lot of time to tell you about that. I'll just tell you that's where we're trying to go, and that we're actively looking for more postdocs. So if you're interested, or more graduate students, if this sounded interesting to you, please think of Princeton. And let me end just by thanking all the people that did the work. Athena Krami did the working memory experiment. Bing Brunton did the original behavior and the decision-making task. Jeff Ehrlich and Tim Hanks did the FOF and PPC experiments. And Mikhail Yartsev did the striatum experiments in the decision-making task. And finally, I'll thank all of you for listening. Thank you.
Time for a couple of questions.
And you showed the rats have the virus devices in there. And I wonder if these virus devices cause stress on the rats, and because it might change the regular behavior of the rats.
So of course implanting something on a rat causes a change in behavior, right? And yes, the rat wakes up from surgery and suddenly it's got this like 30-gram thing on its head. Yeah, it's a little freaky for it. And so we spend a lot of time, so there are two things to say about that. One, this is true of all recording methodologies. And in fact, when we use our regular tetrodes, not only does the rat have something on its head, but during the recordings, it's plugged in. There's a cable that goes to the amplifiers. And having that cable and the feel of that cable, it takes us about a week for the rats just to get used to that. Okay? So both for the tetrodes and for the wireless, we actually first start putting little weights on their heads so they get used to the feeling of something weighing there. And then after a while, they seem to be fine, and they groom, and they seem to be happy. Once they've gotten used to it, it all seems to be fine. The wireless, because it doesn't have the cable, is even better than the wired. So that's another reason why we like the wireless.
We are working on the transcranial focus ultrasound. And
Sorry, say that again?
The transcranial focus ultrasound.
Uh-huh.
And we have also the same obstacles, and listening for what to take.
So we're also using the functional ultrasound, and we now have rats moving around and behaving, and they seem to have gotten used to the weight of that too. So that's one place where it's useful to use rats rather than mice, of course, when you're using something heavy. No, he's asked 3 questions already.
So when you move from the mucimal inactivations to the optogenetics, that was to get at the sort of more acute, sort of faster times.
Time resolution,
If
yeah.
you go the other direction, like if you like our conversation earlier, today,
Aspirate
if you
region.
out the regions are aspirated, could they use other parts of the circuit?
We haven't done that yet, so we don't know the answer to that.
Is it possible the rodents are using a quantitative strategy besides accumulation, such as remembering the site where the last click was?
So we worry a lot about that. So for example, where was the last click? We know that's not what they're using, because that's one of the things that we've checked for. So there's a variety of ways that we've checked for this. So here are the 3 things that have convinced us that they really are using accumulation of evidence. First, the model that we used to quantify the behavior, I said, oh, it has 9 parameters. And it turns out that as you set the 9 parameters to different values, it can actually implement many different strategies. So for example, it can also do burst detection. And it can also do a mix of burst detection and accumulation. And it can do kind of everything in between. So that model alone encompassed a wide range of strategies. And when we fit it to the data, it always does the accumulation strategy. So that suggests out of all the things the model could be doing, it's really accumulation. Then there's the universe of things that the model doesn't do. And so everything that we've been able to think of, we check that, and it's never come out as matching the data better than accumulation. And finally, the one that really led us to drink our own Kool-Aid was you have an idea of what's going on, you make predictions from it, predictions that aren't in the original data. Do they match your data? And it turned out that the predictions were matched really, really nicely. That's if you want the details, it's in figure 3 of the 2013 Brunton et al. Paper. But what we did was take trials where there had been multiple clicks from each side and use those to fit the model. And then from that model fit, now predict the behavior on trials where there was only a single click on one side. Okay? So a completely different class of trials. And it fit beautifully, predicting rat by rat exactly what was going on. So that last one, seeing that predictions from the model are borne out in the data, that's when we thought, OK, we think we can believe this.
Have you tried any primary sensory areas? Because there's been really strong discussions lately about whether or not somatosensory cortex, for example, is involved in simple tasks. So, and people show that sometimes it's not, sometimes it is, right? And we have published a paper recently where we showed that even thalamus in the tactile system has some signatures of these sequential or the history of the choice or the history of the stimulus. So I'm wondering what's your thinking on that?
So we haven't yet, but yes, that's where we're going. A lot of things in my lab seem to be driven by Maya saying, we should go here, and a graduate student or a postdoc saying, no, you're an idiot, we should go over here. And so my bias was towards frontal areas, thinking they'd be more cognitive-y, but a postdoc, Thomas Liu, in the lab said, no, we really should go to auditory cortex and the inferior colliculus, and so he's working on that. So we'll have an answer for you, not quite yet, but in progress.
Okay, let's thank Carlos again.